So let me get started. This session is being recorded. And this is a session titled Aligning Provenance Approaches Across Domains. This is a birds of a feather session, and which is really intended to help people meet each other. Matt, Arifan, could you guys mute both your video and your mic? Good. Good. Thank you. So this session is an attempt to create a conversation about data provenance practices across all scientific domains. In organizing this, we know that different disciplines are in very different places regarding data provenance. Some fields have very well established standards, technologies, practices, and other fields are still trying to get started. And we are also aware that the meaning of data and provenance varies across domains. What we're hoping this session will do is reveal ways that we can learn from each other. To start this conversation, we've invited presentations from 7 people representing a range of scientific research. We've asked them to speak very briefly about current data provenance practices in their fields. And after these presentations, we've invited 2 colleagues who've been active in the computer science and computer technology of data provenance for some time to comment. And then we'll open the floor for discussion. At the end, we're going to ask people if they see common ground that can lead to future meetings, possibly an RDA interest group or a working group if there's something specific we can do. But since we have a lot to do and a number of speakers, I'm going to get started quickly. I'm going to introduce all of the speakers very briefly, and then we'll turn to them in the order that I read now. So we're going to start with social sciences, represented by Arifan Gregory from the DEI Alliance and CoData. Second, astronomy, Mireille Louis at Strasbourg University. My apologies for pronunciation. Crystallography is going to be described by John Helliwell at the University of Manchester. Chemistry by Stuart Schock at the University of North Florida. Earth Sciences by Kirsten Lehnert at Columbia University. Biomedical and Life Sciences by Mark Musin at Stanford University. And Environmental Sciences by Matt Jones from Data One and the University of California at Santa Barbara. And then we will have comments as they find useful by Bertram Luderscher and Tim McPhillips, who are at the University of Illinois. So I remind people to switch off their cameras and mics when they're not speaking. Also, don't click on any red buttons. And finally, you will find at the lower left of the image, there is a button that you can use to expand the image of the presentation while people are speaking. And finally, I've been asked to remind people that this session is being recorded. So let's get started. Arifan, you're up first.
Sure.
You're good to.
Okay. I'm gonna start. Okay. So Lewis Carroll, who you may know, used to talk about
doing impossible things before breakfast. And for me, I'm not an early riser. This is one of those, which is to try to talk about the social sciences. And the first thing I want to do is sort of define how I'm using that term here, because I'm not just talking about a single domain or discipline. It's a group of them, really. And the phrase I like to use is the social, behavioral, and economic sciences. So, and I throw into that category a lot of official statistics and some public health research. So this is a very broad area, but it's an area that has some really common themes, some common elements, because it's all about studying people, and particularly things like behaviors and motivations and beliefs, some aspects of people that are very difficult to measure. And one way that people do that is by asking questions. And so there's a really strong emphasis in survey methodology. So questionnaires get used a lot, and that's a primary source of data across all of these sort of related domains. The other one that's a very big source of data is what I'm going to use the term administrative data, but it's really the trail of data that people leave behind as they interact with the world. And people are born and die, married, divorced, employed, collecting unemployment, going on Twitter, all kinds of different activities that leave records in systems. And to one extent or another, researchers have access to that data. And it's an important source of data for understanding what people think and how they behave. And so those are two of the really common aspects of all of these different sciences when you look at them. What that means is that research findings depend heavily on the methods used for data collection, because the sense you make out of those kinds of inputs is and understanding like what questions were asked is just really important. And so that, I think, informs a lot of how process and provenance are viewed in this area. Another thing I'd point out is that there is a very direct connection to public policy and to government. Now, in the age of COVID, a lot of different sciences are directly connected to policy, as we know. But in the case of the social sciences, I think that's almost always been true. If you produce an unemployment rate, that's immediately politically important, right? And so there's a very direct line there. What are the requirements? Well, I tried to put all of these in one slide, and it's a pretty high level slide, but I think the primary requirement is to document process so that people can understand and use the data. And that includes the data collection, like methods, like sampling and so on, what questionnaires were used, how responses might have been coded, any kind of interpretive aspect of data collection. Also, what we call data editing, which is really the processing, cleaning, validation, imputation, a whole bunch of things that you do to the data to make it fit for analysis. And knowing what was done to the data at that level is quite important. The analysis itself, tabulation, aggregation, there are lots of different methods, but being able to describe that analysis and understand specifically how it was done. Also, there's a class of organizations in the social sciences, data archives and stats agencies, whose primary job is to provide data for secondary use. And what they do very often is to integrate data and harmonize data to make it more useful for researchers. And so you have a sort of a third party coming into the picture and taking disparate data, putting it together, and then offering it to people. And to understand that process is also very important. Data archives also do a lot of different things to data when it's archived and preserved, and that can change the data as well. And so understanding those processes is important. So there's a whole bunch of stuff that's really just documentation. There's also an angle here that if you're producing data, you want to be able to manage processes so that the data you produce is consistent and comparable over time. And so process management in a forward-looking way is an important requirement in this space as well. And I mentioned earlier the sort of direct line to governance and policy. Audit trail can be very important in this world because there can be legal implications for what you do or do not do with data. It's not just that your peers will review you harshly. It's that you may be breaking the law. And so those are things that are important. Interestingly, in this domain, there's a pretty monolithic technology culture, which is to say that for a long time, if you used SAS, SPSS, Stata, or R to analyze your data, you were with 80, 90% of the community. That's not as true anymore, where you have AI and some of the big data approaches, people are using Python. There's a very high degree of literacy in relational technology in this domain, I think. And so that has consequences. Very often what people want, if they want to reproduce your findings, is the data and the processing scripts. And there's not a big concern that the execution environment of an experiment or the way in which those things were used, there aren't a lot of questions around that because it's pretty well standardized from that perspective. The other thing is that process documentation tends to be secondary to data documentation, so that it's viewed as an add-on. And I think this is a very data-centric view that may or may not be merited, and less probably as time goes by. But that is, I think, the case in the domain. I'm not going to go through all of these standards in any detail, but we have a lot of standards. DDI has always engaged with a sort of after-the-fact description of process. And with DDI CDI, we're beginning to be a little more forward-looking, but mostly by relying on other standards like SDTL and VTL or the proprietary processing scripts. The structured data transformation language, SDTL, is now actually being distributed by the DDI Alliance, but it came out of the C2 metadata project, and they're still developing the tools for that. That's an important thing in our world. Prov was another big deal when it came out, and people thought it would solve our problems, but it's not a tool, it's a toolkit. And so we ended up realizing that Prov 1 is probably the best fit for our needs, but Prov is definitely an important standard in this space. In official statistics, you have VTL, which is very similar to SDTL, but primarily focused on validation and reporting of data, which is a big thing in the official statistics community. You still see BPMN for process management, and we have a lot of useful reference models for archiving and for different kinds of processing in the official statistics world and in the data production world and research. So there are a lot of standards. That said, there is not, I think, an agreed community of practice at this point. The demand is very high. It has been for a long time. People know that they need better management of processes and description of processes of provenance. And there have been, in the past 10 years, some very important developments, but there isn't a common solution. We have some standards, we have some tools, but there are still gaps. And like I say, there's no sort of agreed community of practice. The other thing that's happening is that cross-domain approaches are becoming more and more important to the social sciences because we're dealing with grand challenges, those kinds of topics, and you want data coming from different domains. And this means that we need to be able to align our description of process and provenance with other domains. And hopefully we'll find that there are other ideas and approaches and standards and tools that we could use to make a better, more workable approach for the social sciences generally. So that's my presentation here, and I hope that makes sense, George, because big.
Yeah, thanks, Arafan. Mireille, can slides up on my computer, and I will try to share them now.
Okay.
And so, Mireille, you just tell me when you'd like me to
Do you hear me correctly?
I hear you. Can you see the slides?
Yes, perfect. Okay,
Okay.
so we can start if you can pass the slides.
Okay.
Thank you so much. Okay, so I'm presenting today the way the astronomy domain considers provenance and the way we are describing. I'm part of a project which is called the Virtual Observatory for Astronomy, and in this community we are used to share our data. I will speak about this a little bit later, and we are very concerned with tracking the history of all the data sets that we are distributing. So the provenance definitions seen from our side, for us, it's a structured representation, and it gathers the information necessary to capture, to store, and to analyze the history of data production. So we want to know what happens to the data. And then we want to formalize this in a way that a human can understand it. And also it can be used by machines and algorithms so that we can extract this information. So this notion came with the automation of the data production pipelines. And if you can show the slide bigger, you could see the little pipeline that I've drawn here. Thank you. So from an observation at the right, time goes from right to left. Then we generate some images that we call raw images, for example. And then we store calibration. And then these data, they undergo another step of transformation, which is calibration. And then from calibration, we generate some transformed data. And then at the end of the pipeline, we can release data collection and make and send it to the scientist. And this, I think I have something also at the bottom of the slide. Okay. So this is valid, of course, for a pipeline, an observation pipeline, but it's also valid for simulation data, which are also used in the astronomical domain. On the next slide. So when we considered tracking provenance in for the IVOA, for the virtual observatory, we have defined our goals. So in a way, shorting down the picture, and we have 5 points that we want. Five goals that we want to achieve with the provenance that we track. So we want to track the production history and describe all the steps that transform our data from the raw data till the data release. We want to track and record the contact and attribution information. So who has done the job and where are the contact points, the contact information. We would like to use it to locate errors in the pipelines and to help this provenance to define some quality assessment. So this depends, of course, on the scope of each project. And the last goal is that we want to have a structure that we can really query when we are looking into this provenance information. So we really need a model for tracking this information. On the next slide. So this is just to give an idea of what are the data in astronomy. So we have multidimensional data, images that represent objects in the sky and that are can be identified by their type and by their position on the sky. We have maps, we have galleries of images, we have time series. The little diagram shows a pulsar, so the variation of light through time, this curve here. And then on the right, you have, I have some spectra represented from the PESTO program. So this is the variation of flux according to the wavelengths. And all these data, they are meant for long-term preservation. In astronomy, we build knowledge and science from the cross-comparison between observations, and we want to store all the data that have been produced. And eventually, it's also interesting to reprocess old data with new algorithms when it's possible. So the data are all shared in the community after a short embargo period, let's say 1 or 2 years. And then it's shared within a framework, within an interoperable infrastructure that has been designed. It took some time to build this system, It's almost 20 years now. And this system is called the Virtual Observatory, and it was built on standardized formats, which means at the beginning, astronomers agreed to have a FITS format. FITS means flexible, interchangeable transport system, and all the data were combined and presented into this format. So that was a good start to distribute the data. And then to distribute the metadata and every data which is stored in a table format, came another format very much used in the community, which is VO, for Virtual Observatory Table. And this is a kind of XML. And with these ingredients, the project, the Virtual Observatory project, has defined some data models to describe what is an image, how we can identify these coverage in space, time, wavelengths, energy. So this for the data models, and we also have access protocols, so that for one astronomer at one place in the world can launch a query on some archive in another part of the world and retrieve the data he wants to select, he or she wants to select. So all these data models and access protocols, they are described in the link I have put. So now maybe I can skip this one because you will understand that the data files, they are entities, the activities, the tasks, they are activities, and that the agents are most of the time they are some pipeline engineers or the project how you say that, BoF. Or so this is my last slide, and I want to show that we can. So this is just an example of a particular project, which is called the Turrencoff telescope array. And here I want to highlight the different strategies that we have in astronomy. So the CTA project, it's a starting project. So the people involved in this project are currently developing the different code for the activities. So they are building up the processing steps and these are described in the box you see as CTA pipe. And CTA pipe is the software for the pipeline. And in each of the tasks, there is a function that tracks what is done by this task and that generates a JSON file. So in a way, each task is decorated with some kind of message that explain what is done in the task in a JSON file. So this is a first way to track the provenance and to trace it. The second way is to just send messages every time a task is executed. You can send a message and write a line into a big text file, a log file, and then by this way you can read afterwards the sequence of all the tasks that were performed. So this is what we call capture provenance, but inside, which is the provenance is tracked from the beginning when each task is executed. But we also have some other old data collections for which the provenance was stored, but in a very particular manner, in a very particular way, depending on the project. And for this, most of the time, the information is stored as keywords into the formats that also contain the data. So in the FIPS data files, there are two parts, the data on one side, and there is always a header, a text header that can be read easily by a browser. And this is the place where we will find the metadata for the provenance, which I mean for the activities that were launched for the data. So the challenge is to ingest this information, extract it, transform it so that it can be understandable and represented into the provenance data model that we are using, which is shown in the center as and called ProfDM. And then from these two entries, we can gather in a metadata, in a database, all the metadata which are necessary to distribute. So this is the provenance database. And on the right, these are the client aspects. So there are two protocols that can query this database, one which is a kind of SQL-based query, the ProfTap solution, and the other one, the ProfSAP, simple access protocol. It's just a URL and parameters that can send a query to this provenance database. And when you get the answer, you get a file, a prof file, which can be a JSON file, or there are several formats available, VO table, JSON, ProfXML, and then these can be transformed using a derived version of the Prov library that was initially developed for the W3C. And so we can generate from these profiles a graph, and then a human can interpret this graph. So I think I'm at the end of my presentation, and if there are questions, of course, I'll be happy to answer.
Well, I'm afraid that to get through all of the presentations, we can't.
No, that was just backup. I can stop here.
Okay, thank you very much, Mireille.
Welcome.
That was very useful. We have to move on. John, I'll bring up your slides, and if you can open your mic and your camera, please.
Yep, thanks, George. Hi, everyone. Can you hear me?
Yes,
People
I
hear
can.
me? Thank
Yes.
you. So we just need the slides.
Yes, let me get. There you go. Can you see the slides?
Yep.
Okay, good.
So this is prepared jointly with International Union of Crystallography colleague Brian McMahon. We think basically we must be very similar to everybody else, but I suppose the purpose of this session is to show how we're all different. We're very much enthusiastic about linking of our publications, our articles, with database entries and the data sets generally held there. There is a transformation going on towards opportunities with large data archives. The Cambridge Structured Database started in 1967 and the Protein Data Bank in 1971. And our own house journal, Acta Crystallographica, in 1948. So these principles of linking articles with data are very firmly embedded in the community. And the metadata for the experiment, the processing and the deriving of the molecular model and the workflows, there are obviously modern ways of recording those, which are coming in. but have always been followed to give a transparency to what we do. From machine learning, I picked up this term ground truth. And the raw diffraction data, our diffraction images, we are now beginning to be able to preserve. And so any one study, we want to ensure reproducibility, because then, of course, other studies can faithfully try and replicate the original study. And on question of scale, the Cambridge Structure Database now has more than 1 million individual crystal structure entries, and the Protein Data Bank is approaching 200,000. And there are other databases which we've summarized in this article here from a session at the Denver International Data Week, published in Data Science Journal. Next slide, please, George. So this is a highly technical slide, a bit like Mireille's one, but I'll just pick out the skeleton here. So at the left, we have the crystal sample. And of course, this is a distinction between our field and, let's say, astronomy, studying the night sky, which is God-given or some such term, whereas we make our crystal sample. And then we measure the diffraction data on a home instrument or at a centralized facility, which can be synchrotron, X-ray laser, neutron, and so on. And coming in more and more are electron-based measurements, both crystallographic and linked with cryo-electron microscopy. So as we proceed from these measurements at left, we have the data processing, the data reduction, same meaning. Sometimes we say process, sometimes we say reduction. And then the structure solution and the derivation of the molecular model to best fit in as precise a way as possible the diffraction measurements. And then this close linking of journals with databases is very central to our whole operational data and information flow. Next slide, please, George. So, given that the preservation of raw diffraction images is very much a new activity, with the data archives like Zenodo, for example, is really presenting a massive opportunity for us. The bare bones of why we're interested in this is that we can, with new software, hunt for ever weaker signals in the data, and we get more detail in our molecular models from that. We can have benchmarks with improving our methods of analysis with new algorithms and software. We can check the early decisions of the crystallographer on the symmetry of the crystal. There may indeed be multiple diffraction in one diffraction pattern, and the crystallographer analyzes the main one, but now we don't need to restrict ourselves. We can analyze the other lattices. And between the what we call the diffraction spots, we get continuous diffuse scattering, and that tells us about the correlated motions or disorder of atoms in the crystal. So that's coming in increasingly as a structural dynamics aspect. And of course, in terms of this concept of ground truth, the reader and user of reader of a publication, user of the data linked with that publication, they can actually go right to the base of the whole study with the raw diffraction data. And this article, Open Access, with colleagues, explains with case studies the benefits that we can derive from preserving the raw diffraction data. Next slide, please, George. So in terms of file sizes, it's worth just mentioning this. This is for any one crystal structure. At the base of our particular big data pyramid, our diffraction images might total gigabytes or terabytes of data. So they're not small for an individual study. And I told you that the total numbers currently in the databases. So these are a sizable total quantity of data. And when we do the processing, the diffraction intensities come down to megabytes, and the molecular models, what we call the coordinates in three-dimensional space, would be of the kilobytes or 10 kilobytes. And of course, the ground truth takes us from a good degree of objectivity, and the article at the top there is what I would refer to as very subjective. And so this whole linking of everything is so important for the provenance as the basis for trust in what an individual study is. Next slide. So in terms of specifics, I stress the point that, whoops. So I stress the point that we start with our crystal sample. So that is the molecule that we crystallize. We want to record as the metadata the crystal size and also the color. The particular facility that we've used, there are all sorts of details of the instrumentation that could apply to synchrotron, neutron, home laboratory, and also it facilitates global comparisons to have good and detailed metadata. We have the chance to make different manufacturer comparisons. The different software packages that have been developed by members of the community do vary. And of course, this is where it gets a little bit more complex, probably the same for everybody else in terms of the provenance range. You have open source software, if you will, that's what I would call full provenance. You then have non-open source, but free to use binaries. And then you have non-open at all, either of source or binary, until a license fee has been paid. And so this is rather a complex territory. And it's also, to not be prejudicial about it, it depends a bit on the funding of the agencies, whether they're supporting people or not, and a pay-to-use type of methodology may be the only way for survival of a particular researcher. Overall, of course, different workflows can be compared, and that leads to a good feeling for precision and then on to accuracy. Of course, speed can be important, but quality is paramount. And there is, in different stages of what we do, a degree of automation, and that is improved and increased the rate of crystal structure determinations in all of our different subdisciplines. So I hope with that sort of overview, you've got at least a feel for how crystallographers proceed. Thank you.
John, thank you very much. That was really interesting. I'm learning a lot from all of these. Stuart, good. Thanks for popping up on your own. You're on.
Thanks a lot, George. I don't have any slides, but I'm going to give a few comments relative to chemistry. And I have to say that, you know, to try and summarize all of this provenance-related information relative to the whole of chemistry is quite a challenge. And so I would definitely say that first, you know, all the different disciplines vary quite a lot in their approaches to and practices with provenance. And I would also say that what John just presented in terms of the crystallographic data, you know, and CCDC is kind of the exemplar for chemistry in terms of currently, you know, what the best data practices are. And And so as a consequence of that, unfortunately, I have to say that the majority of chemistry is behind in a lot of these activities. But let me just point out where we have some things going and what we're working on right now. In a general sense, and I would also say that my perspective is more from an analytical chemist, which is what my subdiscipline area. So there's a lot of different types of data that are recorded in chemistry. A lot of them are physical property data or chemical property data, and then the additional majority is spectral data, which is typically in support of research as a characterization, or certainly looking for structural information about chemical compounds and materials. When we think about the kinds of data that we put in our research papers, it's the general stuff you might expect to try and contextualize the data that we're presenting. There's a lot of focus on, though, the chemical information. And the best practice kind of historically has been to include something called the Chemical Abstracts Registry Number, which has been minted by Chemical Abstract Service, a subsidiary of the American Chemical Society. And that is now being added to in the literature by the addition of chemical identifiers that have been minted more recently. Things like the IUPAC INCHI Trust INCHI identifier, which is the International Chemical Identifier, which is a code created unique identifier for a chemical structure based off of its molecular graph. It does very well for small molecules, but it still needs some work to be able to represent larger structures. And there are other identifiers that are being created to be able to make sure that we're referencing appropriately the accurate chemical substances, like the SMILE string, or simplified molecular inline encoding specification. One of the things that's very common in research is to make sure that when you are using particular chemical compounds, that you absolutely reference the manufacturer, the catalog number, the lot number, and then if available, you can get access to a certificate of analysis to be able to let the community evaluate the quality of that particular material. In a sense, as far as best practices relative to capturing data, in a sense, we're still back using the traditional research approach of a written laboratory notebook, noting down all of the steps that is done within the research enterprise, including the instruments used, equipment, chemicals, like I said, instrument settings, and visible observations in certain disciplines are very important. We're kind of at a tipping point, I think, right now, in a general sense, being a little behind in the digitization of and the representation in a digital sense of the chemistry enterprise. But I would also argue that it's good that we're maybe behind, because of course the revolution that's happening right now is something that we can hopefully tap into significantly, and we're going down that path already. One of the big areas where provenance is very important is in industry. And there's been a lot of work on what are called laboratory information management systems, which are where major industrial organizations will centralize a data facility to be able to capture spectral data from spectral and chromatographic data from a number of different instruments. And that has evolved into what are called electronic laboratory notebooks, which are in many senses used simply because of the need for legal documentation of different things. So, FDA compliance in the United States, for instance, requires CFR 21 Part 11. And so, being able to record as much of the workflows and the data and the provenance of the data is very important in those systems. Sadly, those systems, as you might imagine, are relatively expensive, and so they have not trickled down really into the academic research community. There are some standards out there that have been around for a while. One is called JCamp-DX, that's for spectral data. And IUPAC currently has a project underway to develop a fair spectral data description, which is clearly needed because of the amount of spectral data that is being generated in the chemistry enterprise. In addition, IUPAC has the thermo-ML specification, which is for thermophysical data, and in that specification, which is XML based, there is a places to be able to put the chemical information, the physical property, the experimental property, and the conditions under which the experiments are done, as you can imagine. So there are efforts like that underway, and we have a lot more to do. One of the things I would also point out is that IUPAC is also working and has worked for a number of years on the development of critically evaluated data sets. And one particular example of that is in solubility measurements. And although that has been published historically, initially as books and now more recently as research articles, there is an ongoing interest in digitizing that because it is in of itself an exemplar to the chemistry community about what we need to do relative to improving the quality and traceability of chemical data. With that, I'll close.
Great, thank you, John. Kirsten, can you go next?
Yeah, here I am. You have my slides, right?
I have your slides.
Okay, great. So a lot, can you just go to the first one? The challenge for me to actually present about provenance in the earth sciences is the diversity of data. And, just listening to the previous presentations, there is a lot of overlap with different other domains in the type of data that are being handled. Just what we just heard about chemistry, about crystallography, about the astronomy has quite a bit of overlap with the type of data that are being acquired in the earth sciences. So I've here just distinguished sort of the two main types. One are really the observations and measurements done in the field and in the lab. And you have anything from the collection of samples and description of structures in the field that are done in a manual way, just observations by your own eyes written down in a notebook, to large-scale sensor networks of observational networks, be it seismology or in the ocean sciences, oceanography measurements, and then the whole world of analytical measurements done in the lab, mostly performed on samples taken in the field, but also experiments that kind of simulate conditions in the field and so on. And then we have the world of the derived data, the models, the simulations, computation that delivers different type of data. Maps belong to that in the earth sciences. And, as in all the other talks that we heard, the provenance here is really to document the history of these data. How were they acquired? What type of instrument was used? How was the instrument calibrated? Or what settings were there? How were samples processed and so on? And when it comes to models, to simulations and so on, it's the code, it's the algorithms that were used, and what data went in into these models and so on. So I think there's a huge overlap with many of the other disciplines. What I noticed yesterday when I was preparing the slides and communicating with colleagues was that for them, provenance was much more focusing on the credit aspects of, who collected the data, who collected the samples, who curates the data, the credit to those who do the data compilations, do data harmonization and cleaning for data products, who curate data, and also to the funding agencies, the credit and the impact measurement and who funded what. Next slide, please. So, and it was fun to have the conversation while the presentations were going on during the session here in the chat about samples, because it's an area that I'm focusing on a lot. And a substantial consideration for provenance in the earth sciences. Where were samples collected? How were they collected? Were they transported in certain ways? There are ways to contaminate, to destroy samples just during transport or during storage, processing of samples, and all that is a really important component of earth science data. Next slide, please. I have kept this presentation very high level because of this diversity of data and clearly being not knowledgeable of everything that the different domains in the earth sciences are doing with respect to data. We clearly follow the FAIR principles, and there has been substantial effort in the earth sciences through COPTIS, the Coalition for Publishing Data in the Earth and Space Sciences, and the Enabling Fair Data project that the AGU has been leading to really ensure proper credit documentation. But the challenge remains in reusability of the data, in that these provenance, the provenance documentation is very domain and data type specific. What is clearly an overarching issue is that meaningful reuse is not possible without that provenance information. Basically determining, allowing others to evaluate, to understand the data quality, to reproduce data, and to be able to compare different data sets or integrate them. And that's a really important aspect. I'm coming from the geochemistry side, and probably the chemists here in the audience can appreciate the problems of different normalizations, different calibrations of measurements, where there can be huge biases depending on which standards are used to normalize data and so on. But on the other hand, the provenance with respect to credit is essential for especially the long-tail communities in the earth sciences to buy into the sharing and reuse in a new culture of open and fair data. Next slide, please. So I just wanted to point to one of the ways that there is convergence in the earth sciences in the way provenance information is captured and structured, and that is the observations and measurements model that exists in many of the data systems that capture observations and measurements in the earth sciences that have the specific concept of process and procedure in it. And I think that's, one point or one structure, one conceptual model to look at if we want to compare different types of data. There is application of the PROF standard as well, but it is fairly rare in my experience. Next slide, please. I saw a question regarding workflows in the instructions to these presentations. Workflows are mostly common in computational approaches in the earth sciences, but provenance of lab analytical data is still captured to a large degree in notebooks. They're becoming more digital, more electronic as we go along, but most of these digital lab information management systems are homemade, customized for different types of data. And as we heard before, the big ones are simply not affordable by smaller labs that perform chemical measurements on samples. The other challenge is that reporting of provenance information is still very inconsistent, mostly in publications following community conventions. We are working hard to create communities that establish specific guidelines that they feel comfortable with, that they can adopt. And coming to my last slide, please. In the facility that I'm heading, EarthChem, which is for geochemical data in the earth sciences, we've seen increased interest and engagement of researchers wanting to establish specific best practices for their communities. So we are providing structures, just web pages for them to communicate what they determine as best practices through a community process. And we are helping them align with these higher level conceptual standards, such as observations and measurements, to foster, to advance convergence in these provenance documentations. I think that's all I have to say. Thank you.
Thank you, Kirsten. Mark, can you go next? And I'll put up your slides, Mark.
Yeah. And
oh, Mark, you've got your own up, so I'm going to set mine off.
okay, are you seeing my slides? Because I'm not seeing them.
Yeah, you won't see them, but we will. But we're not seeing presentation mode.
All right,
you might be showing the.
There we go.
There, that's it. Got
Technology is wonderful. Well, thanks, George. I've been given the task of trying to summarize data provenance in all of the life sciences and presumably also clinical medicine. This is obviously an impossible task because the ground here is just too much to cover. I will talk about life sciences generally. I'll focus on genomics, which is probably what I know best. My full disclosures, I work in biomedical informatics. I'm not a life scientist myself, but I'll tell you what all my friends struggle with. And that struggle really is one where people are consistently worried about the crisis in reproducibility in science, particularly in biology. After the landmark study a few years ago where Amgen showed that it couldn't reproduce many of the classical studies in cancer biology, people began really to worry about what happens in biology in particular. And so there's a strong tradition, fortunately, of putting data in public repositories, at least the data that are of certain kinds, data that are created through large, well-funded experiments. But there's really a weak tradition of providing adequate metadata to describe those experiments. And frankly, when we talk about data provenance in the life sciences, largely that means what we write in our method section, what we remember to put down in our method sections and journal articles, rather than anything more formal than that. So as in earth sciences, the idea of using formal methods to represent provenance is something which is just really alien to most biologists at this point. The other issue, which I think is important in life sciences, particularly genomics, is that people often don't really care that much about the primary data, and that data transformation is really important. And we have lots of really great mechanisms that allow us to analyze data and produce secondary data sets, which are really where our main focus is. And as a consequence, the issues of data provenance are really very important in life sciences. Also, the provenance of the actual samples is important, and there's a whole repository at the National Center for Biotechnology Information in the United States that has a database for the provenance of samples. That's the good news. The bad news is that, like most metadata repositories, particularly in the life sciences, the metadata are not very good. This is just I won't say it's a random sample, but gives you a flavor for how people describe information in biosample, and you can sort of muse on that and know that our team actually has done a formal analysis of biosample, and I can say with confidence that more than 3/4 of the metadata that are supposed to have Boolean values are neither true nor false, and that some 25% of the integers, or at least the metadata that are supposed to be integers, don't parse as numbers, which gives you a flavor for the challenges that people have in dealing with biosample and repositories like it in order to trace sample provenance. We look at what goes on in genomics. Let me just give you a couple of examples. Probably one of the most commonly used tools for analyzing data in biology is BLAST, which is an old tool. It's been replaced by things that are far better, but it's still the most commonly used. BLAST allows you to take sequences of amino acids in a protein, sequences of nucleotides in DNA or RNA, and compare them. It allows you to align different sequences of subunits within a macromolecule and figure out how the elements of one protein relate to another. It's basically the whole basis by which we're being able to identify what are the variants in all of the COVID viruses. And BLAST is a great program. The challenge is that few people really understand the nuances of how to use it. It has lots of parameters. People can change those parameters and get different results. And the two things which are striking and getting a lot of attention in the literature is that many people who use BLAST are ignorant of what those parameter changes might mean for the way that their data get reported in terms of these aligned sequences. And also getting the particular parameterization of the program reported in the literature or in metadata is not all that common. And so consequently, the decisions that people make about how to use the technologies that are used to create secondary data from the primary data are really an area where there's an enormous need to trace provenance, and one which is really not getting the attention I think that it needs. Just give you another example. 20 years ago, there was a big revolution in biology when people recognized that they could create chips where you could put on a chip basically all the genes in an organism and then have those genes followed over time to see how they turn on and turn off in the context of different interventions that one might be able to make. These are DNA microarrays, really changed the whole substrate of how we look at genetics, not as a static kind of area of study, but one where we can actually see how genes turn on and turn off over time. And all the results that were coming out of these large microarrays obviously couldn't be published in paper because there are just too many genes. And the results that were being put online were not always interpretable. And frankly, the biologists themselves got together, created a quasi-standard called the Minimal Information About a Microarray Experiment, or MIAMI, which created a way of saying what should be in the metadata to describe such an experiment so that a third party could understand what was actually done. And this was really a major advance and shows you how a community can get together and actually try to be clear about the provenance of the data on which it operates. And you'll notice that item 6, not well described, but item 6 is the essential laboratory and data processing protocols. What normalization method has been used to obtain the final process data? This is the kind of thing that people recognize for important. The challenge, of course, is that although MIAMI became really important for the functional genomics community, and lots of other areas of science created their own metadata standards, all of these standards, by and large, were textual. They weren't really computable, but they provided a lot of guidance. I think they really represent very important first steps. Within this microarray community, though, one thing that really changed the world was the advent of the gene ontology, which gave us a way of talking about the things that are happening in cells and gave us a hierarchy of information about what happens in cells, where it happens in cells, what molecules are involved. And what happens when people analyze microarrays, what they're interested in is not the thousands of genes that turn on and turn off, but they're interested in patterns. They want to know that genes that are turned on in a particular situation may be genes that are used for metabolizing glucose or initiating photosynthesis or replicating DNA. And so these are really exciting technologies that allow us to take the primary data, knowing what genes are on or off in a particular setting, and to abstract that information into, for example, knowledge that there's a mitotic cycle that's initiating or there's cell division taking place. This is really great. The challenge, though, and this is we get back to data provenance, is that depending on what version of the gene ontology you're using, not surprisingly, you'll be abstracting these primary data into slightly different abstraction categories, because as the gene ontology changes, how you use it changes. And I would argue when you look at the literature, when you look at metadata describing these kinds of experiments, you very rarely see the kind of information that would allow you to know what version of the gene ontology was used to create these data analyses, which are so critical to so much of what happens in biomedicine. So I would argue that the life sciences don't represent a place where formal provenance has taken root and we're taking advantage of some of the great formal structures that people are using now to document provenance. Rather, we are in a situation where biologists do recognize that it's important to be able to document this kind of information. They recognize that metadata are important generically, even though most metadata in the life sciences are not very good. And most important, biologists often are caring not about the primary data as much as transformations of those data. And we really need more attention to being able to document how those transformations are made, how we parameterize the tools that allow us to perform those transformations. Because frankly, I think the life sciences represent a really good use case for why formal mechanisms for documenting provenance can be really very, very helpful. Right now, provenance in the life sciences is really documented in publications, in the method sections, which we all know are getting smaller and smaller, or at least the font size for the method section is getting smaller and smaller, if not the actual content. And the available metadata online, which, as I indicated, are often quite problematic. And with that, let me stop.
Thank you very much, Mark. Matt, can you go next? And let me know whether you'd like me to show the slides for you.
We'll give it a quick try here, and if not, we'll switch to you.
Good.
All set?
Great.
Thanks. So I'm going to talk about the Earth and Environmental Sciences. And like the other presenters, the Earth and Environmental Sciences are really a collection of allied disciplines. It sits at the intersection of the life sciences and the physical sciences, and so covers domains that are quite broad in perspective and coverage, everything from oceanography and atmospheric science to ecology and other types of biological sciences that then get tied into genomics and other areas, as well as even into the social sciences when you look at environmental problems and their application to human systems. So there's a really broad array of different groups, and along with that, there's a long history of documenting the data resources and methods and protocols that are associated with the research in these disciplines. There's a huge variety of metadata standards that have been in use, trying to really understand the data that leads to conclusions in these different disciplines. And so, like the other presenters, I would say there's a long history of people trying to use natural language methodologies and semi-structured metadata standards to document the content and structure and scientific meaning of data and the processes that lead to conclusions in papers. As the reproducibility sort of issues have emerged over the last several years, there's been an increasing call within the literature in the environmental sciences to improve reproducibility and repeatability in ecology and environmental science specifically, that go along with the calls that are in other disciplines and that have been increasingly seen within these groups. So you see, calls in, say, 2006 for increasing reproducibility, and those get translated over time into really practical solutions for how to do that. In 2018, Ben Marwick and others in archaeology and ecology with Carl Bettiger, focused on how to really do that within, say, capture provenance within analytical frameworks like R or other tools that people are using really extensively. These efforts are fantastic, and they really show how researchers can kind of create informal or ad hoc approaches to organizing data and computational processes, but they don't necessarily sort of move to that next level of formality in documenting provenance. And I think that's where there's still a gap in our discipline. So between what people would like to be able to do with reproducibility and provenance and what's being actually adopted and implemented. So we've worked with a number of other folks on reproducibility issues and thought about it in a number of terms. Victoria Stodden wrote a really nice overview of reproducibility and its different components, and can be broken down into different sorts of areas, including the sort of traditional empirical reproducibility of what can be repeated within the lab, statistical reproducibility of the methods and inferencing, and then computational reproducibility and whether you can really understand the computational steps that were taken to produce scientific findings. And so a lot of the infrastructure that we've focused on in the environmental sciences has focused around that third area of computational reproducibility and really focusing on transparency. Like I said, there's a huge variety of metadata standards that are used in the fields. And so one of the challenges is trying to come up with a common vocabulary and a common approach to representing provenance in a way that multiple different disciplines can understand, and the ways to which multiple different tools can utilize to produce systems that are useful for researchers. So we've focused on 4 really core areas that come straight out of the Prov-O vocabulary and approach to doing provenance, and those are really focused on four main things trying to be captured in systems. So versioning is critically important within these systems to understand which version of data and other artifacts in the scholarly ecosystem people are using. So if you have a tabular data set, it might start off as a certain representation in CSV, and then the new version is created later, and that is archived as well. And we need to understand when those multiple versions exist. Derivation is really common. We have lots of complex data products, everything from derived visualizations to complex models and other types of outputs from those models. And those derivations need to be linked back to the original data from which they are derived. And so tracking derivation relationships is really critical for reproducibility in our discipline. It's something that's not done necessarily as often as we'd hope, and that's something that we've been striving for to try to get people to capture. But it's not an easy thing to capture all of the complex relationships among possibly tens or hundreds or thousands of data objects that are sort of involved in a particular analysis. People are also really interested in the transparency of the workflow that is associated with that derivation. So trying to understand what kinds of computational processes were used to generate a particular derived product, and what kind of data inputs those utilize. And so this can be modeled as a sort of traditional workflow kind of conceptualization, where you have a set of inputs, you have a set of processing steps, and a set of outputs, and those can be linked together in a data derivation workflow. And then finally, there's kind of an attribution layer where we're really trying to understand which researchers within the community are responsible for and should be attributed for different types of work, and so that attribution gets tied into the overall model. And so within some of the subdisciplines within the environmental sciences have started to adopt some of the community standards like Prov-O that are used for representing provenance. And so one of those, we've developed a model called Prov-1 under the leadership of Bertram Luderscher and others in the community, trying to focus on how to create an extension of Prov that really can be applied to computational workflows like these that I've described here. And within a network of data repositories called Data One, we've implemented that system so that people can sort of track the detail data inputs, the processing workflows, and the output. So here is a screenshot from one of the data catalogs in the Data One network showing source data that leads through a particular processing step, which in this case is a set of R processes, creates an intermediary data file that's in the middle that then leads to downstream usage and derived data coming from that. In this case, a set of map imagery that's being generated from it in Prince William Sound. So this type of provenance capture is really critical to reproducibility, and I think there's a lot of enthusiasm for it within the Earth and environmental sciences community, but it's not necessarily something that has been widely adopted in formal ways. And so we've also been focusing on how to capture this across multi-generational workflows where one data set might actually come from many others across time and across different institutions and workflows. And so the landscape gets complex, and this contributes to that difficulty in adoption. One of the ways that we've envisioned potentially improving that kind of adoption is to build good tooling around capturing provenance. There have been a large number of efforts in the workflow space to try to capture provenance, and I've been involved with another effort called Wholetail that's really trying to build an environment that allows researchers to work in the computational environments that they're used to. So for example, an R Studio or a Python notebook, when they do that, they can capture the data, the code that's used in a particular analysis in that narrative, and then the compute environment that's involved in that. And so by capturing all of that, we can create what we refer to as a reproducible run. And that in turn can be archived in data archives and used in those formal ways. And so we're hoping that tooling like this within the environmental sciences will really help to improve uptake and adoption of provenance tooling, and then help people to understand and increase transparency, like is the sort of goal of a number of those papers that I mentioned at the beginning. So with that, thank you.
Well, thank you very much, Matt. So we are actually down to the last 8 minutes. So there really isn't time for discussion, but we have another opportunity. This session was scheduled for 2 time slots. There's another time slot tomorrow, which I believe is like 7 p.m. in the US, and I'm not sure what time it is in other places. So what I propose is that that for everyone who is available, we use that time slot to have a discussion about what we've heard today and what we've learned. I've seen some of the presentations ran a little longer than we expected, but I think that there was so much content I didn't want to interrupt anybody. And I also see that there has been a very active discussion in the chat, and there is also an active discussion in the notes document that I think it was Volker has started. Thank you very much for that. We will also ask the presenters to upload their slides to a place where they can be found and link them to the notes document. The RDA organizers responded. I didn't think of this till this morning, but the RDA organizers have given me a link which I'll send to all the presenters where they can upload their slides to Zenodo. So I think that that's the best way we can continue this discussion, and is by I don't want to start anything now, but maybe Bertram or Tim or Simon, if any of you have something that you'd like to say to help us frame how to proceed in the next session, please feel free to open your cameras and mics.
Hello, this is Bertram. One thing I wanted to point out, I think Tim McPhillips put a link in the Google Doc. There's an event that I think is highly provenance relevant where we're looking for real world use cases. I saw a lot of sort of good examples here from multiple communities that have provenance. I'd like to hear from those folks in this event. And again, just find it in the Google Doc. It's called T7. We're emphasizing the importance of transparency, of course, for which provenance is the key technology, but we want to also sort of hear it from the use cases side. In terms of how to proceed from here, I don't have any further suggestions other than if there's a follow-up opportunity to have a discussion, that would be great.
Okay, thanks. Tim, is there anything you want to say, or you want to hold till we have more time?
I would just say, thinking about next time we meet, I always think in terms of who the consumer of provenance is and what questions you would ask of it. And so to me, the need for alignment across disciplines is really, are there questions that researchers would have that span multiple domains? And so my first question for everyone is, what are the questions you use? To me, the use of provenance, most important use, is exactly what John mentioned: trustworthiness. Can someone in the field who's reviewing or looking at the results of in a paper, do they have enough information to trust that the work was done right and the conclusions were reached for good reasons? And so that's kind of where most of that's where I like all these T words: trustworthy, transparent, and so on. So the question is, are those, that's a question. Can I trust the results? But what are the other questions people have for provenance?
Okay, I think that's very helpful. Thanks. Simon, do you want to say anything? Simon, I should point out, is the main organizer of this session.
Only in part. Can you hear me?
Yes.
Yes. Yeah. I've put a suggestion into the chat. In any case, I mean, I think it's a good idea to use the second slot that we have for further discussion. But I do note that it's not very Europe friendly. We held a similar session at the Fair Convergence Symposium, where we really focused on the technical perspective, and what we tried to do in this session was to focus on the domain perspective. As a follow-up to the Fair Convergence Symposium, we had a session a week after to discuss some of the issues in more detail. And I think perhaps not necessarily next week, but in a couple of weeks' time, it might be useful simply to organize a freestanding workshop where we can try and dig through a lot of the fantastic information that came out of those various presentations, and very detailed presentations. I hadn't quite got this into the chat, but it strikes me that the next stage is, okay, we've got Provo, and there are a number of applications of it. What are the points of contact, both in terms of the motivation, the scientific motivation in terms of reproducibility, replicability, and in terms of the way Provo and other approaches can be applied to the various processes and models that we've had. And if we reflect on that, is there basis for improved coordination or at least alignment, coordination, alignment, correspondence in how we talk about provenance, and in essence how both within domains and across domains we improve the practice. Something that came through from a number of the presentations as well, which I thought was important, particularly the later one, well, the earth science one, the environmental sciences one, is how prominent the cross-domain use cases already are in a number of the examples. And so I think there is need to explore this further for alignment across different data types within particular broad domains or cross-domain, depending on the terminology we wish to use.
So Simon, thank you for that. We have just a minute left. Let me make one suggestion to everyone. I put the notes document link in the chat again, and please, if you'd like to be contacted about a future meeting, even after RDA, please put your name and contact information in there. And so we do have to end, and before we're shut off, thank you very much to all the presenters, and thank you to everybody in the audience, and for all of the contributions to the notes and the chat, which we will try to follow up on. Okay, thanks everybody.